Hello, this is Joe Delaney again. In this video, we'll be examining the real math behind SWAN pathway analysis. This is really for advanced users, but it does incorporate both the concepts behind it and some of the math that actually happens. No programming will be introduced here again. So just as a refresher, canonical pathway analysis is fairly simple. You have some pathway of 5 genes. You look at your experimental sample, and there may be 3 genes altered. For most of these analyses, there has to be some sort of background comparison. You may expect only 1 out of 5 genes to be altered, and then some sort of statistical test is used on these 2 populations to determine a p-value. SWAN is a little bit different from canonical pathway analysis in that we incorporate phenotype data. So that is both haploinsufficiency, or some sort of multiplicative gene scoring of individual genes within the pathway, as well as the building of network from something that may interact with other parts of the pathway, such as a kinase. So how do we get the math to be consistent when each pathway is heterogeneous? So pathways do not necessarily look like one another in terms of the actual construction of a network. So for example, we may have a very simple network with just a few genes interacting with one another. The average pathway looks something like this, where about half of the genes encode proteins that interact with one another. And some of the pathways are highly skewed in that one protein is interacting with just about everything else within the pathway. And the pathways you really don't want to analyze are the hairball pathways. This is something like ribosomal pathways, where each ribosome gene interacts with every other ribosome gene. So that really isn't the focus of SWAN analysis. So the question is, how do you analyze all these different network structures with the same framework? All right, well, let's return to biology and think about why we arrived at the answer we did for SWAN. So basically, we want to look at one edge at a time. So for this whole pathway, what does it look like if we're just looking at R and C? Well, let's think about dominant effects here. So if the interaction of R and C are important for forward pathway flux, such as the activation of transcription, you can envision how this particular part of the pathway will work depending on how R and C are altered in your data set. So if R is downregulated, well, the R and C complex does not work anymore. It requires R, so transcription is reduced. The same would occur if C is downregulated. Interaction is diminished, transcription is reduced. And mathematically, these are counted as equal, as long as neither one is haploinsufficient. One aspect that confused us when we were first establishing how to best calculate SWAN was how to treat when both genes are downregulated. And it turns out the one that is best able to predict biological output is when you presume that interaction is not further diminished. And this makes sense. If you require the complex of R and C together, it shouldn't make a difference if it's either R or C, or if both of them are diminished. You can make similar interpretations for upregulated versions of this example, and that's exactly what SWAN does. So this edge is appropriate for many different situations: protein-protein interactions, genetics, or screen hits, and so it seems to be applicable in all cases we've tested so far on SWAN. Alright, how does the math actually work out? Let's say we're looking at just this one edge, and we have data scored here on the top of either -1 on the left side or no change on the right side. And so we have an edge that is counted as -2. And that's simply because we multiply either individual nodes change by 2 because it's within an edge. SWAN tests 2 hypotheses, as discussed in a separate video. One is the suppression hypothesis, and one is the elevation hypothesis. And in the end, we have some sort of output magnitude score, which is an average of these 2 hypotheses. So what each of these do in the calculations is we have to determine the minimum score within this edge, that's a suppression hypothesis, and we have to determine the maximum between these 2 edges, and that is the elevation hypothesis. So the maximum of -2, 0, and a third set 0 is 0 for this edge, whereas the minimum is -2 between -2 and 0. And so that's how we arrive at all these answers here. Now what happens for the many genes in the pathway that do not have edges? Well, let's say we're building a separate network of these two genes. In this case, they do not have an interaction. Then the single gene on the left, which is altered, is not multiplied by 2 because it does not have an interaction. And so the magnitude changes, but the overall estimate, the positive or negativity here remains the same. And the actual magnitude outputs that SWAN uses is that average score on the right side. Alright, so let's switch this around conceptually. So we just apply the same data in reverse, and you can see that the actual average score in each individual hypothesis ends up with the same answer. Now let's say we're doing this for elevation. It's the same sort of idea, whereas in the suppression hypothesis, the minimum is now 0. It's not +1 or +2. It is the 0 within this pathway pair. Whereas the elevation hypothesis now adopts that same magnitude. And the average score is just the inverse of the suppression. Alright, now let's layer in haploinsufficiency. So if one of these genes, say the one on the left, was determined as haploinsufficient, then that incurs a multiplicative score on top of that interaction multiplication already. So that occurs, say it's for a mutation, it would be 10 times by default, or if you have your own custom gene scoring, then that's how it gets incorporated into each individual edge score. And again, each suppression hypothesis or elevation hypothesis will test the paired data against itself and with 0. And that's how we end up with -4, 0, and -2 on the top, where there is an interaction, or -2, 0, and -1 is the average if there is no interaction. And same thing here, if we switch the data around, the magnitude changes, the direction does not. So the magnitude is smaller because the gene on the right does not have haploinsufficiency, and so that does adjust how the edge is calculated. Even though it may be impacting a haploinsufficient edge on the left side, the interpretation by the network in terms of how this is actually calculated does reduce the magnitude. And here again, if we're incorporating both the haploinsufficient gene and the non-haploinsufficient gene and estimating a minimum, well, you can see that even though the data are the same for each side, the minimum hypothesis weights it on the haploinsufficient gene side. So we end up with -4 for this top interaction, -2 on the average score. The non-interacting network does change a little bit here because both genes do have a negative score, so that is slightly increased in magnitude. And so just note that throughout all of this, even though both genes are scored as negative, you might presume that in the elevation hypothesis that the maximum here is actually -2 or -1, but we always test it against the 0 change hypothesis as well. So the max and min can end up as 0 for each of these. And so of course the puzzling aspect is what to do in these situations. So if you have one node that is suppressed and one node that is amplified, this is really where you don't know what is happening in the biology. Either side could be the dominant effect. And so that's why we entertain both hypotheses. So we have the suppression hypothesis, we have the elevation hypothesis, and each of those will derive different scores. And you can see it's equal magnitude on this side, and then the average score ends up being 0. And so if we summarize everything what we've shown here into one of the more simple networks that we can generate, this is something that you can see adds up to an entire network score, not just one edge score. And you can see the one slight change is how that purple edge, the antagonistic edge, is counted. And in the end, the SWAN network score is an average of these two hypotheses, and it would be -2 for this whole pathway. So the end goal is really to create a consistent volcano plot because there's permutations here, we want it to look relatively the same each time we do the calculation. We want to know that our top tip pathways are the top tip pathways. Well, to compare pathways to one another, these network scores would be vastly different depending on how many genes are within them. And so to account for the different size of these pathways, to account for the different number of edges that are produced, then each pathway is lastly normalized to a simulated sample in which every gene has either a -1 or +1 score. And that helps us just create an actual volcano plot which is representative across all the pathways that you analyze. This does remain as an arbitrary interpretation, so it's not like if you get a number of 30, that's a 30% reduction. It will remain somewhat arbitrary, but it is comparable across all pathways, and that's really the important aspect here. So again, to create that volcano plot that is consistent, the only thing that can really ruin the consistency is the shuffling format. We have to do the shuffling of the actual data within each individual sample to arrive at a consistent, reproducible answer that is actually biologically meaningful. And so, depending on how those are shuffled around, we still get those suppression hypothesis, elevation hypothesis, and the average score for each individual shuffle. And we can do between 100 to 1,000 different shuffles for the data. And each of these arrive at the SWAN stands for Shifted Weighted Annotation Network, right? So these are individual shifts of the shuffled data. And so in the experimental distribution, those may all be losses, but in, let's say, a control, and we compare a shuffle to a shuffle. And that is really esoteric as to why this is necessary. When we were first designing this software years and years ago, we didn't think this was necessary. But when we tested a variety of different data sets with very different structures, we found this to be absolutely critical so that SWAN could be applied to just about any form of data. And so the idea here is you actually take your randomized data and compare it to another set of randomized data. So each time you have your sample shuffled, it is actually shuffled twice. And so this is actually the distribution of the randomized shifts that are observed. And again, we do this 100 to 1,000 times for each individual sample. And so we've discussed, there are different network distributions for each individual pathway. And the goal is we want this SWAN pathway analysis to be applicable to any of these different types of networks. And so I just wanted to show you how these shuffling aspects actually turn out in terms of the distribution of how the shifts are produced. And so this is a ranked plot from left to right, from the most shifted negatively to the most shifted positively on the right. And the reason we do this is in the end, this is a Wilcoxon rank sum p-value. And so this is the distribution, which is analogous to how that p-value is calculated. And so in the experimental set for that very simple network, you can see most of those shifts for the experimental data are negative, with a few of them just randomly positive. Whereas the shuffle to shuffle comparison can be anywhere in that negative range to the positive range, and then the average of it does center around 0. Now we take those same data and just test it across the different networks, and we see that the ones with normal interaction look fairly simple. Fairly similar. The magnitudes are slightly different, but the distribution shape remains the same for both the experimental and the shuffled. And that's also true for this highly skewed prolific interactor type of network. And that's really the goal, right? So we can use SWAN to analyze any type of network data. We don't like the hairballs, but it is potentially usable for those as well. So the p-value, as I just mentioned, is a Wilcoxon rank sum. So one group always has to be this experimental group, with the SWAN shift scores associated with that experimental group. And so the control comparison may either be SWAN shift scores from a shuffled random control, which just means you take your own data and you shuffle it into those two pairs and compare against those random shuffles. So in this case, you would only have experimental data, and then the background would be strictly computational. However, in most biological cases, you do have a control data group. And so this control would be like another experimental group where it is compared to shuffled random data. So you can have either just a strictly computational background or this control background, but either of them do require that background shuffling of the genes to determine how this network's expect value is calculated. A paired Wilcoxon p-value is calculated for each shuffle, and so that can either be that control data to the experimental data, or it can be the shuffle data to the experimental data. And the reason for this is that p-values need to scale with sample size, not the number of randomizations. The median of all these randomized p-values is the actual output p-value of SWAN. So if you do 100 shuffles, then you get 100 Wilcoxon p-values of each of these shuffles. And so the actual p-value that is output is the median of all of those randomized p-values. Why? Well, the stability of the p-value due to heterogeneity in the shuffling process. And I'll just give you a few visual examples of how that comes out. So, shuffling does cause noise. And so here what we're representing is 10,000 shuffles, or a really extraordinary amount of shuffles on cancer data. And this is ranking pathways from the lowest p-value, the highest significance, to the least significantly altered p-value of 1. And so in 10,000 shuffles, you would hope that you've arrived at an answer that will not be changing much. Well, what does that happen if we use less shuffles? As we want to do in actually analyzing our own data, we don't want to take more time than is necessary to analyze the data set. So with just 25 shuffles, our answer is very different from what this 10,000 shuffle may be. Our top hits, the top 25 here or so, still remain suppressed. They still have a low p-value, but a number of them look like they're false negatives now, where the p-value has reached 1 for some of these otherwise suppressed pathways. That is just highlighted here. Each red bar here would be a potential false negative call. And actually, it doesn't matter how many permutations we do. Even if we go all the way up to 10,000 again, then we may actually get some false negative calls. And that is just because of the way these data sets are enormous, and these networks can be very heterogeneous. So how do we get around this? This is, again, really esoteric, but for those who are interested, we use an ensemble majority vote method. And this is used for a crystal structural determination, but they actually ended up being really useful for this network pathway analysis. And so an examination of, let's say we only have 3 different shuffled cases, and we end up with this overall SWAN network shift. And the average shift for the first shuffle is negative 101, the average shift for the second shuffle is 32, and the average shift for the third simulation is 36. Well, even though the magnitude of those 2 positive scores is not as high as the magnitude for the negative score, if you average this out through a mean, it would be negative, right? But the actual majority hypothesis utilizes each of these as individual tests. And in this case, we actually have 2 out of 3 tests saying that the most likely hypothesis for this is an elevated pathway. This is an example of just a 3-vote majority used here. And so when we apply that on the same scale from 1 to 25 different votes, we can see that this is really what reduces that false positive noise. And so when we use 25-vote majorities across our total permutations, So, for example, if we have 100 permutations, we would do 4 of these majority vote determinations, and we see on the right side of that 100 votes, very few false negatives come up in the analysis. And that is true for the 200 permutations, 1,000 permutations, or the 10,000 permutations. So this is really helpful in terms of reproducibility of a pan-pathway SWAN analysis. So just looking at these different simulations that we've performed, for average users, we recommend a SWAN setting at 25 vote majorities with 100 total shuffles. That makes it a very short analysis time with minimal false positive or false negative occurring. There are limitations to this. The lack of modeling a distribution makes SWAN much slower than some alternative pathway analysis methods, such as the hypergeometric methods often found in Go analysis. And this really is not designed for very low sample size experiments. So if you only have 1 or 2 samples, well, this is a distribution. So Wilcox and rank sum does not work at all for 1 or 2, and it doesn't work very well for even sample sizes of 3, 4, or 5. Of course, the strengths of this are simply that you arrive at the biological answer, which is very likely to be positive when you follow it up in future experiments. And of course, the shuffling of data across networks mathematically enables very different network structures to be analyzed by a single consistent method. So you don't have as many assumptions that you need for your input data. You don't have this certain distribution which it must match. And so this is why SWAN is flexible for us to integrate different forms of data, such as DNA, RNA, and mutation data all together in a single network analysis. The magnitude of the SWAN shift is even more stable than this p-value, so I showed you how sometimes you get false negatives, but really the magnitudes come out about the same every time. And the stabilization approach has really resulted in less permutations necessary to arrive at your ranked pathway confidence, and so this does speed it up. One aspect of the software that we haven't really discussed much is that gene level data can be utilized rather than the entire network level data. And that's how we've gotten around this sample size limitation, because we recognize many of these big data sample sizes are at the three sample level. And so to get statistical significance with that, you can still get Wilcox and rank sum comparisons, but instead of using one network per sample, you use all the genes within that pathway per sample. And so that does mean your p-values will be more significant for larger pathways, but at least you can get to some statistical significance for these lower sample sizes. And we've really collaborated with a number of investigators and used enormous cancer studies, and we've been surprised at how the top results have been really consistent, reproducible, and most importantly, have been able to be chased down with shRNAs or overexpression later and produce a phenotype. And so, taken together, I hope you've learned a little bit about how the math works without going into the actual programming. And if you're interested, please do look at the actual published coding and see how this works at the mathematical level through vectors and matrix manipulation. Thank you for your time, and enjoy using SWAN.